Hello again, Year 9, and welcome to the Walking Talking Mock Part 2, where we'll be continuing on with going through the paper that you were assigned on Monday. So yesterday, for Part 1, we did questions 1 to 4, not including 4. Today we're going to be doing 4, 5 and 6. Like yesterday's lesson, you won't be able to do this unless you've actually sat the paper that was sent to you on Monday, so please make sure that you do that first and then tune into the part 1 of the Walking Talking Mock, and then it'll be this one. Okay, so question number 4. 15% of your questions across all of the biology, chemistry, physics papers, all 6 papers, will be based on science skills. And this kind of thing is a classic science skills. It's one of the required practicals because you're looking at osmosis in potatoes. And also it's asking you about the technique, why they're doing certain things, and then calculating from that. So we're going to go through. Don't be put off by science investigation pictures or where it's got more information on it. Just read through it, take a pen or a highlighter to your exams and write stuff on the questions. You do not get marked down for writing on the questions or making notes around the outside. So question number 4. Some students set up an experiment to find the concentration of sucrose solution in potato cells. The students used discs of potato cut to the same size and weighing approximately 10 grams. The discs were put into each of 5 beakers. So you've got beaker 1 that's got distilled water, so that's pure water, 0% sucrose. Then beaker 2, we've got 10% sucrose. Beaker 3 is 20% sucrose. Beaker 4 is 30% sucrose. And beaker 5 is 40% sucrose. So the first question says, after 2 hours, the students carefully dried the potato discs with paper towel before reweighing the discs. Why did the students dry the potato before weighing it? And it's worth 1 mark. So don't overcomplicate it. Just give a simple reason for why they dried it out, and that should get you the mark. Now the answer that they have suggested is that it doesn't affect the mass when weighing it, or something to do with the water on the potato or the solution on the potato would affect the mass when put on the scales. So anything to do with that having an effect on the mass would be the correct answer for that. Okay, so 4.2. The students calculated the percentage gain or loss in mass of potato. The results are shown in table 1. So it doesn't on here give you the percentage of sucrose in each of them. So if I was in the exam, I would think about adding that onto it because it did give us that at the very beginning of the question. But what it does give us is the final mass, the initial mass, and then the percentage change. Now, last week I said to you that out of the biology, 10% of the questions are going to be math-based, chemistry 20, physics 30, and a really common maths question, or something it might ask you to work out, is the percentage change in something. So we need to be able to work out that percentage change. Now look at the number of marks. It's 3 marks, so it's not going to be as straightforward as the ones that we did last lesson. There are going to be a few stages to this. Each stage will be worth a mark. So even if you don't get to the end answer, if you are showing you're working out, you pick up marks for each correct stage. So I would always suggest, and I know they nag about it in maths, always show you're working out, because you do pick up marks even if the final answer is not correct. So the first thing we need to do is find the difference between the initial mass and the final mass. So if we're working out the difference, we just need to subtract. So the first stage, of which you get 1 mark for doing, is 10.4 minus 7.3. So that's finding the change. And you should get that as 3.1 grams. So that will be worth 1 mark in the exam. So that's a third of the marks already, just doing a bit of subtraction. The next thing we need to do is divide by the original mass. So we need to take our 3.1 and we need to divide it by 10.4. So pop that into your calculators, and I said last lesson, always take a calculator to all of your science exams. If you put 3.1 divided by 10.4, you should get 0.29 or 0.3, it doesn't matter which one you use. And that again is worth another mark. The last thing we need to do, because we're getting it into a percentage, is times it by 100. So if we take the 0.29 and times that by 100, we make it makes 29%. Now, if you did it as 0.3, it'd be 30%, and you would still get that final mark. So any numbers 29 or 30% as your loss of mass will get that final mark. So that's 3 marks, and it's just following that process. If you're asked to work out percentage change, first of all find the difference between the numbers, then divide by the original, and then if it's in a percentage, times it by 100. Those are the stages you need to do. Okay, moving on. Okay, and the last question for number 4. Predict the concentration of sucrose solution in the potato cells using the results in table 1. It's only worth 1 mark, so it's going to be something that they think is going to be pretty straightforward. This is where it would have been useful to have put the percentages of sucrose. Beaker 1 was the water, the pure water. Beaker 2 was 10% sucrose. Beaker 3 was 20. Beaker 4 was 30, and Beaker 5 was 40. So we need to look at the results and see where there's no net gain or loss. And that's going to be happening between Beaker 2 and Beaker 3, because Beaker 2 is gaining, Beaker 3 is losing. So it has to be somewhere between there. So they will accept any number between 10% and 20%, because it's going to be between those two. If there's no net or gain, it'll be because the concentration is the same as within the potato cells. So that's telling us what the concentration is in the potato cells. Quite often I've seen this as a graph, and it would be where the graph goes through the x-axis, because that would be your 0. So because it's only 1 mark, it's only going to be something simple. So you've just got to point to where it is. Remember, this is the purpose of the practical, to find the concentration within a cell. And that's looking at where it's not gaining or losing anymore. Right, let's move on to the next question then. Question number 5. Some scientists investigated the rates of absorption of different sugars by the small intestine. In one experiment, they used a piece of normal intestine. In the second experiment, they used a piece of intestine poisoned by cyanide. Cyanide is poisonous because it prevents respiration. Now, that's going to be an important sentence later on in this. So, as I say, you do not get marked down for making notes on the question papers. So please feel free, if you come across something that you think is important, then highlight it. So we've got our table 2 results. We've got the rate of absorption of glucose, galactose, xylose and arabinose in normal intestine, and then the intestine poisoned by cyanide. And already you can see some of them are pretty much the same and some of them are vastly different. 5.1. Name 2 sugars from table 2 which can be absorbed by active transport. Now, all of this relies on that sentence. Cyanide is poisonous because it prevents respiration. So if you are stopping respiration, then you're stopping active transport. You have to be able to make that link between them. It's worth 2 marks. So if you look at the table, the ones that have been greatly reduced in the cyanide one is going to be glucose and galactose. So you'll get 1 mark for each correct one of those named. So as I say, is that link between respiration and active transport. Okay, 5.2, and I've put the table back on this slide. Use evidence from table 2 to explain why you chose these sugars. So this is worth 4 marks. So we're going to have to use the data from table 2. So that means picking out important bits of information to show what you're trying to say. And you need to explain. So you need to say why it's happening. Now on this one, I've actually included the mark scheme from the exam. So you can see that they split these bigger questions. So it's normally any that are worth 4 to 6 marks, but they split it into different levels. Now for level 2, the top level, you need a detailed and logical explanation which identifies the evidence from the table and links it to the explanation. Logical links are made and scientific terms are used accurately. So if you're able to use the information and use it to explain why, that's your top marks. Level 1, which is 1 or 2 marks, discrete relevant statements are made. So you've put good science in there, but nothing links together. It doesn't flow on. The logic may be unclear and links may not be made. So underneath, they'll have indicative content. So this is the kind of thing you could be talking about. So the evidence is the absorption, there's less absorption in the cyanide intestines compared to when there is no cyanide. They're absorbed faster than the other sugars. Why? Active transport needs energy. Less or no energy is available because cyanide is present. So there's less energy and no respiration. So that's the kind of thing they'll be looking at. So if you put in there that, back to the table, so if you put in your answer that glucose reduced by 2/3, or there was a big difference between the amount of glucose in the normal intestine and the poisoned intestine, and the same for galactose, because it requires energy to move it in active transport. The energy is transferred in respiration. That's going to be all of the marks. So think about how you're answering these, because it's not just going to be 1 bit of science, 1 mark. You do get put into these different levels. So you've got to make sure that you're fully answering the question, linking together the active transport and the data from the table. Right, the last question of question number 5. All of the sugars named in table 2 can be absorbed by diffusion. Explain how information from table 2 provides evidence for this. And this is going to be worth 2 marks. So we need to be able to say what it is in the table that tells us that all of them can be absorbed from diffusion. So the answer for this, your first mark, would be that all of the sugars are absorbed in the intestine poisoned by cyanide. Even when there's no respiration. So that'll be your first point. So you can see on the table that even though the intestine has been poisoned by cyanide, which means that it won't be able to do any active transport, there's still sugar moving through. So that's what that first point is. And then the second point is saying that diffusion doesn't need energy. So if you said that diffusion doesn't require energy, so it's still happening, that would be your second point. Right, question number 6. Bone marrow contains stem cells. Explain why bone marrow can be called a tissue. And it's worth 2 marks. So the first thing we need to do is think about what a tissue actually is. What is the definition of a tissue? And it's a group of cells, a group of similar cells. So that's going to be your first mark. What do stem cells in the bone marrow do? They make blood cells. So if you said it's a group of similar cells, which work together to produce blood cells, that would be the full 2 marks. If you said it's a group of cells which make blood cells, then that would be the 2 marks as well. So for that, we need to know what a tissue is, and which comes up in a different chapter, and we need to be able to say what the stem cells actually do in the bone marrow, and that's making blood cells. If you named a blood cell, like red blood cells or something like that, then that would still give you the second mark, so you would get 4 marks for that. Right, 6.2, and this is the last question on this paper. And this one is giving us a chunk of information, and a lot of people will see this and automatically switch off. But you've got to keep going on these kind of questions because they are going to be big chunks of marks. Not doing them can be the difference in a grade. So the first thing it asks you to do is read some information about stem cells. So stem cells are used to treat some human diseases. Stem cells can be collected from early embryos. These stem cells have not begun to differentiate, so they could be used to produce any kind of cell, tissue or organ. The use of embryonic stem cells to treat human diseases is new, and for some diseases, trials on patients are happening now. Stem cells can also be collected from adult bone marrow. The operation is simple but may be painful. Stem cells is bone marrow mainly differentiated to form blood cells. These stem cells have been used successfully for many years to treat some kinds of blood disease. Recently, there have been trials of other types of stem cells from bone marrow. These stem cells are used to treat diseases such as heart disease. Evaluate the use of stem cells from embryos or from adult bone marrow for treating human diseases. You should give a conclusion to your evaluation. And this is going to be worth 5 marks. Now, the important command word is evaluate. So we need to give pros and cons of both, and then we need to give our opinion. That's the conclusion. It won't always tell you to put a conclusion, so please make sure that you do whenever doing an evaluation. Okay, so here is the mark scheme. Now, if we have a look on here, it says for all 4 marks to be awarded, there must be at least 1 pro and 1 con. So we need a pro and a con for embryonic stem cells, a pro and a con for adult stem cells. That'll get you 4 marks. The 5th mark is going to be a conclusion based on what you've just written. So which one you think is best. So if you have a look in the second column, it gives you the pros and cons that it talks about in that text, plus other ones that you might have learnt in lessons. So you need to have 1 pro for embryonic stem cells. So you could say they can be used to treat lots of different diseases. It's plentiful, so it's easy to get hold of them. You could say it's better than wasting them. And you can say it's painless. Those are the pros. And you only need to have 1 of those. For the cons, you can say that it is harmful, or there is death to the embryo. It's untested or new and unreliable. It might not work. And the embryo can't be asked. There's some ethical issues involved in that. So if you added 1 pro for embryonic and 1 con for embryonic, that's 2 marks already. For the adult bone marrow stem cells, Pros are there's no ethical considerations, it's a quick recovery, it's relatively safe, it's been used a long time, so it's well tried and tested, we know that they work. So if you've named one pro for adult bone marrow, then that's your third mark. Cons, any operations, there's risks of infections. You can say it only produces a few types of cells, so it's only a few diseases. You can say that it can be painful, so it might deter people. So if you've put one of those cons, then that's a fourth mark. And then if you've given your opinion based on what you've written, that's going to be your fifth mark. So if you said that embryonic stem cells will be the best because they can treat such a wide variety of diseases, something like that, that's going to be the fifth mark, giving your opinion. Okay, so that is it now for the walking talking mark. We're going to be continuing this cycle for the next few lessons, and we're going to be looking back at another chapter that we covered during lockdown. So what I would like you to do is just send me an email at deatonS@knoleacademy.org. You can put S Deaton, it doesn't matter. And just give me your total out of 35. Okay, so just send me an email or do it on Show My Homework, what your total was out of 35. I hope you found it useful. It is very important to know about those command words and looking at what you're doing and what you should be doing. So hopefully that was a useful exercise. Have a lovely day. And next time we're going to be looking at the next chapter. So take care until then. Goodbye.